Many animals need to stick to substrates. They need to glue themselves in place, or they need to produce mucuses that may need to be more liquid or more solid gel-sol transitions. I'm going to start by talking about how a mucus, a related molecule, can make those transitions. Imagine a mucus that has many strands. Imagine a mucus with many strands that I'll simplify to single strands that is highly hydrophilic, and so it's absorbing a lot of water, but it's sticky. It's sticky to itself and water, so that gives it its mucus-like sticky character, but it's quite fluid. You could easily pull on it and pull it apart because the interactions between these long strands are all with hydrogen bonding with each other or with water. Now let's imagine that there are negative charges spread along these strands. This would have very little effect on the structure of the strands because the negative charges would all wind up with a hydration shell around them. And again, that would contribute to the viscosity, but it wouldn't cause these strands to be unable to separate from each other. Let's imagine what would happen if these were either inside an animal or were molecules that were secreted to the outside and we allowed calcium entry. When we write calcium, we often write Ca plus 2, but of course the charges are not all on one side. It would be more accurate to write Ca plus plus. So if we release calcium into this medium, anywhere that two negative charges are near each other, we'd form a bridge. The bridges would be constantly shifting because there's hydrogen bonding as well as the ionic interactions, but the calcium would tend to greatly stiffen this, and with enough calcium, they would resist pulling apart. There are echinoderms that apparently use a system like this with connective tissue, extracellular matrix that is quite fluid, a mucus-like set of molecules, when the animal is relaxed, but upon disturbance, the animals release calcium that forms these calcium bridges and makes this connective tissue very rigid. When the animal is relaxed again, when predators are gone, they can pump the calcium back out and regain a very fluid, liquid-like structure of the body. What about secreted molecules and glues? Let's imagine a cell that is releasing vesicles of a mucus-like substance, lots of strands with branches, and there are positive and negative charges distributed unevenly, so that scattered in all of this mucus, there are positive and negative charges. The substrate is irregular, and it has regions that are positively charged and negatively charged, distributed at random. We would expect, as this fluid mucus that has no covalent bonds between these long strands, the strands would match up. So anywhere where there was a positive charge, it would tend to go near negative charges, and anywhere there was a negative charge, we'd expect positive charges to be nearby. So these strands are moving, and they're spontaneously winding up near wherever there's an opposite charge. That will enable this mucus to stick quite well to the substrate, and if it's also sticky to the animals, to the cells of the animal moving by up above, by hydrogen bonding or other interactions, then the mucus is going to form a sticky layer underneath a snail or some other animal that tends to stick the animal to the substrate. How would we convert that to a glue? There are a lot of different mechanisms that can convert molecules like these into a glue. Many aren't known. This is an area of biomimetic design, trying to understand biological glues and use them to make better glues for humans. I'll just toss out one possible mechanism. If this mucus has arranged itself with the positive and negative charges near each other, then it's going to adhere quite well to the substrate. Now, water molecules can get in the way, so this would still be fairly fluid. Water molecules could interact with that positive charge and the negative charge and wind up pulling this away. Let's add molecules being secreted that are small molecules that can form a covalent bond on either side. An example of this are quinones, a carbon ring with a reactive group on each side. So let's add a bunch of these that can form cross bridges between different strands. Now what happens is that the different strands I've shown here will begin to be connected. And the more of these covalent bonds form connecting the strands to each other, the more rigid this whole structure will get. If there are structures like hemidesmosomes, protein structures in the cells that are up above here, that have protein components that extend out into extracellular matrix, and if those wind up being connected, perhaps by some of these same quinone cross bridges, what we'll do is start forming a stiffer and stiffer mass here. What was fluid like a mucus is now starting to set like a glue because of these covalent bonds. The glues can set by shrinkage, which makes them even tougher. That happens because these crosslinks can happen only when part of one of these strands gets near to another. Think about two strands that are distant from each other. They're flexing back and forth just by random molecular motion. And there are water molecules between them. Consider what happens if just by chance this strand gets close enough to the neighboring strand that a quinone can make a link between them. That happens again. The strand that was over here with a lot of water in between has now been moved, been pulled up here, and all of this water that was here has been forced out. The water that was between these strands mostly no longer fits. This has become dry. And if we took many different areas with individual strands that get closer and closer, pushing out water, over time we're going to shrink this whole area of glue smaller and smaller, pushing out water. The water that was interfering with these ionic interactions is being progressively pushed out. The whole mass is getting drier and drier and shrinking. That makes these ionic interactions stronger because water is no longer present to disrupt those bonds. That can make these biological glues astonishingly strong.